So I'd like to open this session for this Wednesday afternoon lecture series. Today we're going to be hearing from Tobias Meyer, and I am very pleased to have the opportunity to introduce Tobias because, in my view, he's one of the most creative and original scientists in the field of cell signaling in the world. Dr. Meyer's research over the last 2 decades have focused on the rules that govern how cells integrate inputs from multiple receptors, how they then compute this information, and then how they make decisions about the key cellular outputs, such as cell migration and proliferation, which is what we're going to be hearing about today. Now, one of the things that really, I think, is a very distinguishing mark of Dr. Meyer's is that his research is integrative at many levels. So he constantly is integrating new approaches from mathematical modeling to large-scale screens to new fluorescent imaging strategies in everything that he studies. Now, Dr. Meyer began his career in Switzerland, where he was born. He was a student originally at the University of Basel, where he studied mathematics and biology. He then got his master's in nuclear physics at CERN, then moved to the University of Basel again to do a PhD in physics. And it's only after that that he then sort of came back to biology, integrating his physics and mathematical expertise, and ultimately ended up at Stanford starting in 1995, where he has been since. And he is currently now the Mrs. George Winsard Professor in Cell Biology in the Department of Chemical and Systems Biology at Stanford Medical School. And I want to just thank Dr. Myers for joining us today. I think we're going to have a very exciting talk on shotgun siRNA perturbation to dissect growth factor-triggered proliferation and migration signaling systems. Tobias.
Thank you very much for inviting me. Can you hear me in the back? Yeah. So I would like to, so my laboratory originally was interested in focusing on particular signaling systems, and we have been working on calcium and phosphoinositide signaling and became increasingly aware that it is very difficult to understand what calcium actually is doing in cells without taking a broader view of this, because there is more than 100 known calcium binding proteins that are present in many cells at the same time. There is many more calmodulin binding proteins that get regulated by calcium calmodulin. So it's often difficult to really try to make sense of, if calcium goes high in a cell, what kind of response actually is elicited after this happens. So based on that, we started really thinking more about how can we look at signaling maybe from a bit bigger perspective, because this didn't only happen in calcium signaling. This also happened in other fields, like the RAS pathway suddenly became very broad. PI3 kinase signaling become also multifaceted and very complicated. So how can we go back and take something as complicated as these signaling networks that we now know exist and try to make sense out of them and integrate them in a way that we can actually understand and work with? So this is the problem, and I want to maybe start with the kind of challenge of what we are facing here. When we look at humans, we have approximately 10 to the 13 cells or so. So that number is quite high. So if I remember that, I usually think of the American debt, federal debt, which is approximately $10 trillion. So that's that number of cells we have in our body. It consists of about 200 cell types. So that's how many things we have to understand, and maybe there's more than that. And there is at least 4,000 of the 23,000 or so proteins in the human genome. 4,000 are dedicated to transmit information from receptors to particular effectors. So that's kind of the outside challenge. And you might think, given that problem, it is very difficult to get to the bottom of that. And what most of us have been doing over the last many years is to pick particular signaling proteins or small family of signaling proteins and try to understand how they work, kind of to resolve structures of them, to try to understand what different amino acids and regulatory features are, reconstitute them in vitro, and so on. And this is probably not so fruitful in understanding how these signaling networks work, because we don't know how to translate these in vitro measurements into in vivo data in a real cell. So this is another way of looking at this problem, that a typical cell in our body has a number of inputs that are typically present at the cell surface. So we are sensing nutrient and stress if there's enough amino acids and glucose, for example. We sense the hormones and growth factors that might be present externally. We sense, and there's signals emitted from cell interaction sites like cadherins and others. And cells also sense interactions with extracellular matrix, typically mediated by different forms of integrins. So this is kind of the input level, and this is, in addition to that, we also have a number of cell cycle checkpoints where cells sense things like whether the DNA is damaged and many other things. And the same types of signaling proteins are often utilized starting at these kind of checkpoint signaling places inside the cell. Then you have signaling components, and their task is to integrate this information and then to make particular decisions. And today we will be focusing only on two of these, on how cells might generate collective migration behaviors and on how they decide to either divide or stay in a quiescent state. So to give you, again, some numbers here. So there is only a limited number, we think, of actually important mediators of these many signaling proteins that kind of transmit most of the information that comes from these receptors. And these include things like the second messengers, cyclic AMP, cyclic GMP. IP3 calcium is a very famous one that we have studied a lot in my laboratory, also the PI3 kinase pathway. You have the RAS MAP kinase pathway and many others like Wnt signaling, STAT pathway, and so on. So these are kind of modules that often consist of something like 30, 50 or so genes that regulate different aspects of these signaling pathways. And then there's also a separate type of modules that often people describe in the literature. They are associated with particular functions. Let's say there is a specific fraction of the signaling proteins dedicated to regulate whether a cell will divide or not, and this has been termed the restriction point at the G1S phase cell cycle. And then you have a migration control module that includes proteins like RAC and others that make a cell migrate or not. So you're having this kind of a modularity that will help and facilitate our understanding of these much more complex systems. And since there's only a limited number of them, there's hope there that by finding these modules that we can learn something about very basic and key features of these complicated networks. And the first part of my talk will be focusing on how to learn something about modules and modularity in signaling system. Just to give you the complexity here, we think there might be only in a typical cell something like on the order of 500 or so proteins that are important in sensing different signals, so quite a bit of them actually. Then you have maybe 40 of these modules that transmit the information from these inputs to function. And then fortunately, most human cell types only have very few functions. Like a pancreatic beta cell, its main purpose, as far as we know, is to secrete insulin, or many other cells have a primary role in just contraction or in sealing a two-dimensional sheet. So this is actually also a very hopeful thing, that by having per cell only a number of outputs that we really want to and should understand, we can also kind of reduce the difficulty of many of these complex networks that I mentioned before in my talk. So the second reason why I think this problem is tractable has to do with simple information considerations. So when you think about a receptor that is feeding a signal into a cell, there aren't actually so many different things that can happen. So at the end, the cell will create a certain output, and it turns out that pretty much everything that we have so far described in signal transduction can be reduced to some very simple circuits that exist inside the cell. One of them is a feedback type circuit, and the other one is a feed-forward type circuit. So for example, we have recently in a review and in papers characterized some of these possible motifs that exist in cells, and there's very few of them. And by characterizing them, we can really learn most about what is really key in terms of the time course and the kind of modeling behavior of particular signaling pathways. And this includes the very important negative feedback, which is often used to create adaptive behavior. Positive feedback can in many systems be used to create bistability. So we'll be seeing in the cell cycle example that I will be going into later that there is a positive feedback, we believe, that creates a switch so that the quiescent cells either fully starts replicating all its DNA or not. And then we also learned that in many cases that you have dual and multiple positive feedbacks, and these have the interesting feature that they can be much more robust. So by linking up more than one pathway, you can create systems that have better kind of properties in terms of being resistant to changes in protein expression level and noise inputs. So these two concepts, feedback, feed forward, and on the other hand, this modularity, we believe can be a guiding hand in trying to elucidate how these complicated signaling networks function in cells. So now the problem is that experimentally, ideally what biochemists and many cell biologists wanted to do in the past is isolate important machines that the cell uses, like motor machines and others, and try to in vitro reconstitute them and learn how they work from that approach. And this has been very successful in many cases and led to many exciting findings. The problem is with the signaling problem that you cannot ultimately do that. There's things like localization that are important, things that diffusion, and many aspects that are very difficult or impossible to recreate in vitro in a test tube. So we have to understand signaling networks by going back into cells or maybe organisms and trying to learn something about these signaling networks by perturbing these pathways with different methods. So we have really two things that we can do. We can perturb these pathways and we can observe. So perturbation of cell signaling systems have recently mostly consisted of genetic type approaches where people looked in model organisms, knocked out genes, and then saw changes in behavior of signaling systems. And the immune cell signal transduction pathways have been largely elucidated with these type of techniques in terms of identifying many of the important players there. Also, siRNA recently has been important, and the main part of my talk will be focusing on that, how this is useful to learn something about these systems. And also, my laboratory has been very interested in developing chemical tools, which has the advantage that they're very rapid, and rapid perturbations are very informative in cell signaling because it allows you to either increase or decrease steps deep in the signaling network, and if you trigger those, then you can look what is downstream or upstream of that particular step in a signaling network. So these are the key pathways that we have, and more recently, several groups have developed also technologies using light activation of enzymes, which I think has a lot of potential in analyzing these behaviors as well. So then the second strategy is observation. And initially, if I would have given this talk 10 years ago, I would have been very positive about being able to come up with live cell biosensor strategies to do all this. In the meantime, I'm a bit more hesitant, and I think the live cell biosensors are a challenging thing, and we still have to, and I will make some comments afterwards about that. In many cases, antibodies are useful. Mass spectrometry, mRNA profiling, and other types of readouts are also very useful to read out what is happening within a particular cell. And the other thing we learned over the last years that in many cases it is very useful and impossible to understand a signaling system without having single cell data. So in many cases, signaling pathways are so heterogeneous between different cells that if you do a bulk analysis using mass spec or so, you can't really understand many of the key aspects of it. So in many cases, it is necessary to go back to single cell using either live cell biosensors or antibodies. So how now to to practically go about this. And I want to make a few comments about these tools that life cell type biosensor tools that are very useful and the most powerful to do this, and then some comments about perturbations. So the field of life cell biosensors really started with the invention of calcium indicator by Roger Chen in 1980. This was a very poorly performing indicator at that time, but it really kind of it showed the path to many people of where things could be going if we would have ways to monitor at the single cell level what is happening with a particular signaling step. In this case, it was calcium that went up in response to receptor inputs. I think the second big breakthrough in this field to allow us to get single cell observations of signaling processes came with the advent of GFP. And Shimomura, Chalfie, and Chen really made the key contributions here in developing GFP into a useful research tool. Our own laboratory was interested in many different applications of these concepts. And for example, we found ways to target calcium indicators subcellularly to membranes or to the nucleus and do localized studies of calcium signaling. Also a development of a photoactivatable GFP to measure in the RAS signaling pathway some diffusion type things, processes. Then the huge breakthrough was the development of calcium FRET biosensors and of translocation biosensors. So our laboratory worked mostly on developing tools to measure phosphotyrosine, diacylglycerol, PIP2, PR3 kinase signaling in cells. So for example, here you see an example with the AKT pH domain we developed, where you see that PDGF growth factor stimulus will lead to the translocation of this domain from the cytosol to the plasma membrane as an indicator of PIP3 generated at the plasma membrane. So this is interesting because it gives you two pieces of information. The first piece is that PIP3 went up in that cell, and the second one where it went up. So this has been, for example, used in the work of many people to look at gradients of PIP3 and others in cells. And since then, a whole flurry of applications of this were led further to the development of sensors for cyclic AMP, RAS, protein kinases, cell cycles, and so on. And I think this field is still in an early phase, because for most signaling proteins we still don't have biosensors that really are functioning in an inert way in a cell and can be used also in whole animals. So I think there's still a lot to be done, and I think that's where also the topic of my talk today comes in, that I think we still are not capable to do this in a systematic way, and much more will be needed to do that in the future. So the second comment I wanted to make is about perturbations. And ideally, we would like to have for every molecule that's important in the cell a specific inhibitor. So we would like to have small molecules that can specifically bind to only one protein kinase, inhibit it, and then you can study what its role is at a given time in a particular situation in a tissue. So what we have been interested in is coming up with a way to activate a larger group of signaling proteins like RAC, CDC42, PR3 kinase, and other enzymes, using a translocation strategy whereby we express the cytosolic protein enzyme activity and then pull it to the plasma membrane using an analog of IRAPs. And in a number of papers, we worked out a way to very efficiently remove almost all the proteins from the cytosol and pull them to the membrane to get a very sharp increase in the concentration of this activator peptide or enzyme activity at the plasma membrane, which then can be used to start a signaling process. I will just give an example of that, where we use that to uniformly activate in a neutrophil PR3 kinase. So before that, we knew in monocytes, neutrophils, and other cells that when you apply a uniform gradient of F-methyl-Phe, they start to polarize, have a gradient in PIP3 to the front, to the back, which then leads to a migration of this cell, which is also called chemokinesis. In this direction would be here, where the AKT pH domain is high. So this led then to a lot of models, and Carol Parent here and many others have developed some key concepts in this field. And we were interested to test now this simple model. What happens if we use the PR3 kinase and not the one that's activated by normal G protein coupled receptors, but we just uniformly raise PIP3 in cells and tested what happens to cells. So we blocked G proteins using chemical inhibitors and then pulled a peptide that can bind to PI3 kinase alpha and beta to the plasma membrane and ask the question, what happens in the absence of F-methyl-Phe to an unpolarized cell if we do that? So what you see here on the left is two cells that got transfected with this peptide in the cytosol initially, and it's a peptide that can bind and activate PI3 kinase alpha and beta. These are nuclear markers here, and here's the time in minutes. So we're now pulling these peptides in these two cells to the plasma membrane, and then you will see these are nuclear markers here, so these are other cells that are not transfected in view here. So these two cells here will then show a very dramatic change that the statistical analysis showed is equivalent to F-methyl-Phe mediated polarization and migration. So these cells, in the same way as cells that are exposed to G protein coupled receptors, still polarize and migrate. So this allows us to say that the mechanism that leads to the polarization of PI3 kinase and to driving this migration response has to be downstream of G proteins, and it's not upstream of here. So these are the types of experiments you can do with these approaches that you couldn't do with genetic or RNAi type approaches, because you wouldn't really know whether you induce this in a timely fashion. You wouldn't know over which time frame this happens, and so on. Okay, so now I want to come to the heart of my talk, which is about this strategy that we have been working on for quite a while now, trying to use systematic perturbation based on RNA interference to make sense out of signaling systems. So my laboratory and many others have used RNA interference in the past as a discovery tool. So what we wanted to do is to knock down particular genes that either going through a human RNAi library or to targeted libraries and find out which of particular genes are important. And then we did traditional kind of focus experiments on these proteins, trying to understand them. So for example, we used that to discover an ER calcium sensor, STIM1 and STIM2. So these are two isoforms that are present in the ER. And when ER levels drop, they form oligomers, go to ER plasma membrane junctions, and activate calcium influx. So these kind of approaches are useful for us discovery tools, but we wanted to use them for something different. We thought if we have 50 or 100 genes that influence a particular function of a cell, then we could use this as a perturbation set and try to learn something about what each of these 50 or 100 genes are doing in a particular cell type. So with this in mind, what we then typically are doing is that we are taking a specific project as a process in this cell, like cell migration. And the first one I will be showing in the second example, it will be about this proliferation assay. And we ask which genes that either we have from discovery type experiments or from literature that we can identify perturb this cell function. And then we go in and we measure different important signaling pathway steps, monitor them, quantify them in an automated way, and then try to characterize all the genes we have into what kind of roles they have overall in this signaling system. And the first example I will be showing is on collective migration, where we use this approach to identify a few core functions that collectively migrating endothelial cells have in organizing their migration behavior. So we think we can reduce all what they do, these endothelial cells, to 5 key processes to understand their collective migration. And then the second one, I want to point out a different application where we use the RNAi strategy to learn something about feedback and bistability in a control system. So this is the problem we were interested in addressing. When you grow endothelial cells on a 2-dimensional matrix, they form a tight sheet. They're impermeant to solutions that you add on the top. They don't get to the bottom. And when you make a scratch and open up in other ways, an open space in the middle, and you activate growth factor stimuli, they will move into that open space and in a fairly slowly, maybe at 10 microns per hour. And by going there, they will then fill up that space and ultimately reseal this space. Sometimes this is also called a wound healing assay. We see it more as a way to study growth factor triggered collective migration. So the question now here is, what steps are between this growth factor receptor and the triggering of this migration response? And there could be all kind of things happening. It could be cell division, there could be the importance of this, could be to polarize cells or to induce migration and so on. So we didn't really know that when we started this. So the tool that was essential for doing this work was this 96-well Delrin tip scratcher. So we call this a scratch tool, which is basically these 96 spring-loaded tips can be used to make uniform scratches in a 96-well plate, so which is very handy because now you can transfect 96 RNAi and each one sees exactly the same scratch in there, and then when you do comparisons you can do this very quantitatively. This is very difficult. We tried this to do with many other approaches with robotics and we couldn't get this to work as nicely. So we think this is a quite useful tool for this simple assay type thing. And you see already here in a plate where we use now in different wells in this 96-well format that some wells now close faster than controls, some of them stay open, indicating that here the RNAi would have suppressed this migration response, here it would have induced it. So this was our core assay, and we used that to screen a library and identify 150 different RNAis that are affecting this behavior and made some controls to be sure that these are the right ones. And now we wanted to come to this simple problem. Can we somehow now reduce this complexity and take these over 100 genes and assign them to specific functions, so that this collective migration is probably relying on all kind of different behaviors, and we would like to break it down into specific functions that might be happening. And I just show you how this movement looks like in a movie. And what we are doing here is we put in a nuclear marker into each cell. So you see here a sheet of cells, so this is tight. So here you would have tight junctions and coherent junctions connecting these cells together. So this is an intact sheet. And in this case, we have eliminated the cells here in the middle, and it's open here. And each of these nuclear-steins now can be tracked with automated software over time. So this can also be done in 96-well format. We can move from one well to the other and keep monitoring all 96 as they move into this open space. And I just show here these two wells monitored in parallel. So one of them scratched, the other one not. Here you see over the 16-hour period they move into this space, and you can see that each cell here is moving. And here, to our surprise, we find that even in the absence of FGF, actually these cells move already quite rapidly in these sheets, and it doesn't require any growth factor signaling. So there's an inherent motility that these cells have that single cells don't have. So if we isolate these cells, plate them alone, they don't move. But if they're in the sheet, they actually move in a collective fashion somehow, because you see here this kind of streams and other things are very apparent in this. So now we're going in, and for each of these 2,000 cells or so, we track them over time, and we make them these kind of things, these maps here, where you see neighboring cells that move in the same direction have the same color. And they have kind of a mean velocity, and we can do now an analysis of the coordination of cells, where we look, if one cell goes into this direction, how likely it is that a neighboring cell goes in the same direction. This is this coordination coefficient that tells us over which distance these cells move in a coordinated fashion. They also have random movement, and they have some coordinated movement. And I will come back to that because we think this is important for retaining the integrity of these sheets, that they have this ability to move in coordination. So we measured 4 parameters. We also found that our RNAi in many cases affected the density of the cells in these sheets, and the density is very highly correlated with the migration. So we measured that, and we measured this coordination. We measured the basal migration in the velocity in the absence of growth factor, which is this inherent ability of cells to polarize and migrate. And then the growth factor triggered directed migration, where these cells collectively move into the open space up and down into this area. So when we do this type of analysis, and because this part is published, so I want to go through this quickly and come to the unpublished part, we can do a separation of genes into those that are affected by one of these pathways and those that are not. So here, for example, we look at the cell velocity in the confluent sheet, and when we look at the GF receptor knockdown or an adapter FRS2 or P10 or these phosphatase here, you're seeing that there's very little effect on the movement of these cells in these confluent sheets. But there is a big effect on the normalized sheet migration into this open space. By separating all these different things in this way, we can make these kind of trees where we sort out which genes are mostly, and I should say here mostly, so they all have maybe some effects on each of these parameters, but they have a predominant effect, these 9, on proliferation. Then 91 in this kind of branched would have mostly an effect on migration that's independent of proliferation. Then we can break it off again into a directed migration part. So that's where the FGF receptor here would be, and some of these other genes. And then these other ones where it's affecting the velocity already in the absence of FGF. So this is not related to FGF signaling. So this falls out. And then we can do further separation into those that just affect primary just motility. So in here you have RAC, you have CDC42, you have RhoA and some other proteins like that. In the cell-cell coordination, you have beta-catenin, you have caterins, and some of these other molecules. So we can break now these genes, and some of them have known functions and make sense, and others have unknown functions, and it's a useful tool maybe to be guided towards investigations of what they might be specifically doing in that context. But instead of doing that, I want to now focus on a different type of logics we are using this for, and I want to just go through that here. So based on that, we basically found that the FGF receptor primarily acts on this directed migration where cells at the boundary, and we actually could show it only acts at the cells that directly contact the open space. All other cells are insensitive to the FGF receptor activation in respect to migration, so we can't see a change in their parameters. So only these boundary cells respond, and they direct cells up here. And then these cells in the back coordinate and probably follow these cells, and I will show you in the following experiments how we determined that some more. And then you have this basal migration and polarity module that allows cells to polarize in sheets and migrate, which includes many of these things. And somehow these different processes work together to give you this emerging sheet migration behavior. So now what we are trying to do is, is that really predictive? So this is the nice thing, because we have now all this single cell data, we can ask the simple question, can we now take simple assumptions about what is happening with these modules and plug it back in and recapitulate these migration behaviors that is fairly complex that we see within the sheet and into the open space. And what we find is that one thing we need absolutely is this ability of cells to, in an autonomous way, polarize and be motile if they're inside a two-dimensional sheet. So we assume that this is a kind of an intrinsic property of cells, and without knowing all the details about the molecular mechanism behind that. So this is a kind of a necessary requirement for any kind of quantitative model. The second one I mentioned before is that these boundary cells here are polarizing and moving into the open space. And this polarization, what we call pioneer behavior of these cells, is causing the directed movement of the sheet into that open space. So we need that term. And then we actually found that in order to explain what's going on inside the sheet, each of these 3 terms is necessary, otherwise we can't describe what's going on. One of them is very logical. There has to be some kind of a repulsive term that explains that nuclei don't fuse with each other because they still have membranes between them, so that's very apparent. And there has to be an adhesive term. If we don't have that in there, the sheet falls apart, and the modeling would say that, and it's also kind of logical. And then we need, these 2 of them would not explain this coordination we actually see in cells, so you need for that some kind of a lateral drag whereby these catenin junctions here can remodel in a way that they exert a lateral drag force onto the 2 cells, so that when one of them moves, it has a slight pull on the neighbor that helps reorienting it. So the logics here is that we think there is an autonomous basal migration state, and then there are these 3 different steering terms that help orient these cells in a direction where they will go. And you have this kind of independent term where cells sense open space and then also in a stochastic way reorient themselves towards this open space. So there's several points I just want to make very quickly that we think are important here. So one of them we see on the neighboring cells, and this has been reported in other cells before, these cryptic lamellopods, whereby actually these cells in sheets have little lamellopods, they stick onto the neighbors. And we think this is important for this polarity and migration behavior because they kind of push the other cell off the surface as they move forward and do that. And this is happening at the bottom area towards the exocellular matrix, and the cadherin junctions, when you look in 3D, are above that. So all the cell-cell junctions are kind of above, and we call this a turtle model to best explain that, where you have these bottom cryptic lamellopods that lead to polarization and migration, and then these sides on top. So that's one part. The other part is, which is kind of difficult to understand, how actually cells that have tight junctions and these cadherin junctions, how can they reorganize? And here we have transiently transfected cells, four cells that touch each other, and you see, as you monitor them, they actually are staying connected, but they reorganize their contacts, and we just kind of made a little sketch here to show that. So this cell here, if it moves in this direction, will ultimately form a contact with this one here and break the two contacts with these cells up here. So this is how you have to imagine how these cells and these sheets can be dynamic while at the same time retaining tightness for liquid exchange between the bottom and top. So we can now take these simple terms and just say, can we now just make a kind of model where we say that we have these steering terms where neighboring cells exert some steering onto an autonomously migrating cell, and we have this directed steering of cells here. And indeed, we can then reproduce the behavior in the absence of FGF. Without that term, it wouldn't move into this open space. And if we add FGF, it would move into this open space. And I just showed this in a movie where we can then map which forces are dominant or which steering terms are dominant at different times, and you see these show up as arrows, and this allows us then to measure these reorganizations as they progress upwards. Okay, so we can now reproduce most of the behavior of these cells using these simple models, and then we tried a couple of tests to test how far we can go with this. So one of them was this crosshair model, where we did crossed removal of the cells using, again, these Teflon tips, and then looked what would the model predict and what would our experiment predict. And the model would predict, against my original intuition, that it's the cells on the side here that win out because there's more of them, and the way how the sheet will reorganize is that it will mostly go in this and this direction and not in this direction. I thought it would go this way. I would have thought this is correct and not this one here. And this is indeed predicted by the model. So it has some predictive power and may explain some not so intuitive results of how these sheets are behaving in culture. A second type of thing that it recapitulates is this streaming behavior. This is a short simulation of that, and if you take the drag term out, then it wouldn't show any streaming here. And then the main point I want to make here is that when we actually looked at small holes, and we now removed the drag term in the model, we could see still that these small holes could still be closed. So without this, if there's no FGF and no drag, there would be no movement into the open space that I showed before. So FGF is required to populate this open space. So we thought also small holes could only be closed if there is a FGF or some kind of a growth factor stimulus present. But this model actually predicted this inherent ability of cells to coordinate with each other, to migrate. This coordination behavior is sufficient to generate small hole closure in these sheets. And so we tested that in an experiment where we made a big hole with a scratcher and then a small hole up here with a little needle point. And then we asked what happens to this small hole here. And even in the absence of FGF, we find that this hole is closing. We expected it to close with FGF, as we would have expected the cells to move into the open space. But it also closed the small hole in the absence of FGF, and it did not move here into the open space, arguing that this really is a plausible mechanism. So this model is also useful to explain some kind of interesting new physiology in this system. So to summarize this main part of my talk, I would like to say that we resolved from this perturbation experiment that the main purpose of the PI3 kinase RAS and other growth factor related pathways that showed up in the screen is to induce directed migration of these boundary cells with very little or no role in doing the regulation of cells in the back. And we also resolved that this coordinated migration behavior is somehow related to this adhesive and drag type of terms. And I didn't have time to talk about the repulsive thing, which is important to create more uniform distributions of these cells. And otherwise, you would have more non-uniform distributions. And all of these things require this autonomous migration, which is kind of the core and most important module in the system that allows these cells to be motile and dynamic over time. So in terms of genes, this allows us now to make assignments and say that these regulatory genes that we identified and maybe that other people are identifying can be assigned to particular modules, which simplifies the understanding of this kind of complex architecture where you in the beginning think PI3 kinase might affect all of these things. This would argue has a main purpose here, and then you can look at other regulatory mechanisms, how they control particular aspects of this complex behavior. And we think this is kind of, this modularity logics is kind of one way how one can combine these systematic perturbation approaches with monitoring key steps in a complex regulatory system and then really work out what the different parts are doing in a more systematic way than was possible before. So I added this second part to my talk, in part because I think this is another very important concept, I believe, in signal transduction. And this is this concept that we have to understand the kinetics of signaling and what kind of input-output behavior we get, and the role of positive and negative feedbacks in regulating these control processes. So quiescent cells, and most of the cells in our body are at any given time quiescent. So we're talking about fibroblasts or so, which probably spend most of the time just waiting there for some kind of a signal to be able or forced to divide and to somehow maintain tissue or repair tissue if needed, or in hepatocytes in the liver and in many other systems. So these are kind of unipotent stem cells in some way, one could call them that, because they still have the capabilities to divide, but they are kind of in a quiescent state. And the question really is, how do they get out of this quiescent state? And we know that growth factors are uniformly important in doing that and triggering this exit. So this concept of the restriction point has been introduced by many groups, which argues there is a point of no return where cells either go forward and replicate and duplicate their DNA, or they continue and they keep repressing each of the 30,000 or so origins of replication that might become active and activate somehow a DNA replication event locally somewhere in the chromosome, which would ultimately make the cells defective. So anything in between is not good, so you either want to do 30,000 on or all 30,000 off. So how do you build a system like that has this kind of reliability? So people believe that there's these mitotic signals, and there's some redundancy in here, but in most cells, this is CDK4, maybe 6 kinases that bind to cyclin D, become active, and then trigger an initiation of RB phosphorylation, which then leads to a de-repression of a transcription factor, E2F, which starts then CDK2, cyclin E activation, and ultimately DNA replication. So our question here was, what is the feedback architecture of this? Can we use these kind of RNAi approaches? We know a lot of these regulatory proteins. Can we learn something about that? And what is actually this restriction point? Can we define that in more molecular terms? So this was then our idea. We would measure 2 parameters here. We would measure this trigger phase. We would find a way to measure the phosphorylation of RB, which we found with inhibitors for CDK4. If this is inhibited, then we get a significant reduction in phosphorylation of RB, which we don't get with CDK2 inhibitors, arguing that these sites are fairly specific for CDK4. So this activity readout is shown here in a single cell assay again. So we measure the intensity in each of these cells. And then as you stimulate cells, the RB phosphorylation goes up. BRDU, which is a measure of DNA synthesis, which is often used to monitor S phase, gives you then also an induction down here. So we can now use this and measure in each of these well, again in a multi-well formats, doing different RNAis, what is going on. So to give first the overview from the biochemical data, when you do this and measure some of the believed key regulators. So there are these CDK inhibitors, P27 and P21. They go down with time as you stimulate with growth factors. Then you have P53, which goes initially a bit up, but then again down as the cells enter this critical phase here that is associated with the restriction point. Cyclin D starts at nothing, goes up. This CDK4 activity comes up also, maybe a bit later, and I will show data on that more quantitative later. The E2F will go up and so on. So you can do this kind of analysis. Some things are regulated up, some are down, and this kind of fits into this scheme that people have described before in other cell types. So we're using here a primary cell culture because we didn't want to use a transformed cells, which some people would argue have defects in some of the key regulators of this pathway. So these are a neonatal foreskin fibroblasts that we're using for that. So these P proteins are basically inhibitors of these cyclin D, CDK4, and CDK2 cyclin E complexes. So the way how we went about that now, we did single cell analysis where we tracked over time what is happening to the build-up in this readout for CDK4 activity, PRB phosphorylation, and here we monitored the synthesis of DNA going from 2 copies to 4 copies by just measuring the fluorescence intensity of all the DNA stain we can do. So this is a fixed cell experiment here. So as we stimulate, we start seeing out of this 1,000 cells or so here, some of them start going up here at 3 hours, actually very few, almost nothing. And then only at 9 hours, suddenly you start this burst of activity where cells start showing phosphorylation of these sites. So I should say that CDK4 is already pretty high here, and there's no activation of this site, which was very intriguing and is important for what I want to say later. As they go above a critical level, you start seeing DNA synthesis, and then you start seeing DNA synthesis also in the buildup of more DNA per cell. So one can now analyze this type of thing by making these bars and say how many cells are at a given time above this threshold and do these types of plots. And I think they're very useful because it shows kind of, gives mechanistic insight what might be happening here without having to do live cell imaging and being able to use native markers in cells. So this shows basically that up to 6 hours, though even though CDK4 is very high, and there's CDK4 activity should be very high because cyclin D is very high, CDK4 is there, we see no activation of this site. So this was kind of an intriguing finding, and then it suddenly builds up, and then interestingly, then it has a steady build-up, which you can see as the increase towards the right, that when you look at relative levels of this PRB build-up, it's about the same distance to the next one. This is consistent with what is often found in positive feedback, because once you trigger a threshold, you just kind of linearly build up the activity over time. I should say the turnover of this phosphate is 3 minutes, and you see here a build-up in activity over 8 hours. So if we do now knockdown of different RNAis that accelerate this step, we see with P21, which accelerates the build-up, it now happens after 3 hours. I don't show the raw data, but believe me that it looks the same, just shifted to the left. With CDK6, it shifts to the right and will only much later start the build-up with still the same time from showing the initial phosphorylation to the higher phosphorylation. These are kind of hallmarks of positive feedback processes, but we didn't really know from that what the positive feedback might be, but it looked like there is a positive feedback driving this system. And I should say that BRDU in all these cases is correlating with a very specific level of RB, which you see better in this plot here. When we plot the relative RB phosphorylation as a fraction of cells that start entering the cell cycle, you see a cooperativity of 9. So whenever you see something so tight, it means that something important is happening in a cell, because it means there's a very tight control of that. We know that the basal activity is somewhere down here, and at least a 6-fold activation is happening. When you think about this cooperativity, it means that you get a 10 million-fold increase in the chance of showing DNA replication with only a 6-fold increase in CDK4 activity. So these kind of cooperativities are very meaningful because they can exert a very strong suppressive activity on a pathway. So based on that, we thought most likely there is some kind of a positive feedback here. There's a variable start time, then CDK4 cyclin D activity starts kicking in very rapidly, but only slowly builds up over time, and then after like 8 hours reaches a critical level where replication starts. So how does this delay, how is that really generated? And the and I shouldn't go through all the measurements that didn't work, but when we looked at P21, which is one of these CDK inhibitors, we found now, and this is something you wouldn't have found with a bulk cell assay. So if you look at the 3-hour time point here, where the P21 is maximal initially, you wouldn't see in this region any cell that has any CDK4 activity. So red means a lot of cells, so this is maybe 2,000 cells here plotted in a diagram that shows where most of the cells are here and here at this time after 24 hours actually. But when you look at the medium level, it is at 3 hours up here, at 9 hours it's down here, and at 17 hours the median level now has switched over here. So this is the path the cells will take. They will first lower the P21 down here, and then they will build up CDK4 activity. So what is really limiting, and what this argues the CDK4 activity and why we see this delay, is that P21 has to be taken out of the way. And the interesting part about that, this seems to require cyclin D. So when we knock down different things, so we can knock down P21, now this whole bulk here moves to the right, you can knock down other things. But most interesting, when we knock down cyclin D, the P21 degradation doesn't happen. And we actually think this is important by cyclin D. CDK4 is binding P21 fairly tightly and can release some of the CDK2 activity, which then enhances the degradation of P21. That's our current model, which is based on some data I will have only very quickly time to show. I don't want to go over time. But basically, we see a second drop. So this is the first drop that I want to mention. So P21 has to go to 10% for CDK4 and presumably CDK2 activities to significantly start and build up over time. So they go from 3% activity to 100%. And as it builds up, there's a critical time when you start seeing DNA replication. So this is a different way of measuring that down here, where we look at the PRB, the phosphorylation status, and then we look how many cells have started synthesizing DNA. And only when this P21 drops to about 1% do we start seeing an increase. So we think there's two steps here. So first you have a drop in P21 to 10%, and this would be impossible to see in any kind of Western analysis because you couldn't see a drop of P21 that far down if 30% of your cells don't trigger this mechanism. So I just want to touch on that, and I will not have time given to go into that, but we have evidence from drug inhibitory studies using 4 different types of inhibitors that the positive feedback that is needed for P21 degradation really comes from CDK2 activation that has to happen to a very weak degree in order for this inhibition to be released. And these studies led to evidence for at least 3 types of positive feedbacks in this system that are engaged during this time window that builds up the CDK2 activity in here, and many other mitotic stimuli, growth factor stimuli that are initially triggered, like the production of cyclin D and P27, are independent of these positive feedbacks. And then we can do again here this perturbation analysis, and I don't want to show you this very long, but basically when we now take over 100 of these RNAis that perturb this process, they all fall on a line. And this is now the kind of approach that I think are useful to exclude or include positive feedback as a possible mechanism. So if you have a case like that where all your RNAi affect at the same time DNA synthesis and this RB phosphorylation, which happens 8 hours earlier, then you pretty much can be sure that the 2 of them are very tightly coupled. And the only way how we know that things can be coupled so tightly is positive feedback. So we think this can be served as a very compelling argument that 2 distant processes in a cell that are temporally distant and also mechanistically different are coupled by positive feedback. And we have some other evidence for that it appears to involve the assembly of replication complexes that has to occur in order for this degradation to be initiated. Okay, I just want to show the second type of evidence is that there is also a very tight correlation between P21, which is this inhibitor, and the degree of RB phosphorylation. And this is what happens in the medium concentration measured of of P21 and the medium level of RB phosphorylation after 24 hours. And this is when you knock down P21, it all shifts up here, you have less P21, and you have many more cells that enter the cell cycle. Now if we go again and we take 200, over 100 of these RNAis and we knock them down, they all fall on a tight curve. And this basically says that for most of them there's a super tight correlation, so all what these other pathways are doing is appear to regulate the level of P21, except of a few things, very few things, that, and we took here 3 different RNAis targeting all cyclin D1, and 3 different RNAi targeting CDK4 and 6. And what you see here is that these RNAis surprisingly are outside of this loop, arguing that, and they don't really have time into that, but you can actually use this now to come up with what kind of a parameter is really the key parameter for triggering this switch. And we think it's simply the difference between P21 and the cyclin D CDK4 that is present in these cells. And since P21 binds and activates a lot of things, this is kind of a very tight limitation. So there's a few things that affect cyclin D, many more things regulate P21, and this is the critical term that decides whether a cell or not engages in that. So we think that this is the trigger point, and then you engage this bistable switch that has this kind of a control structure with 4 types of positive feedbacks. Given the time, I don't want to say much more, but we basically have now a trigger mechanism by which the difference between the P21 level and the cyclin D level and CDK4 activity level is measured by cells. This is the trigger point. When this is engaged, this whole system here is ramped up and runs during 8 hours until DNA replication will be triggered. And we were also able to link this back using simple modeling to this restriction point, which we actually think happens just about at the time when this RB phosphorylation starts, because the cyclin D stability is so that it's still enough of it around until it enters the S phase and can commit to the cycle. And you just would see that here there's a bifurcation point, and when you cross that, then cells will just commit and enter the cell cycle. So to summarize this, we think there's a trigger point, a bistable switch that's based on both CDK2, CDK4, RB, and E2F. And the most important part is that this barrier P21 has to be removed in order for this to go forward. So to summarize these findings, that my main point here was that these types of perturbation approaches, when combined with multiparametric analysis, are very powerful to give you a modular structure of a complex regulatory circuit. And I used this motility system to make that argument preliminarily. And for the feedback loops, I used the system with the restriction point to show how this is useful to identify how feedback loops might be wired. In our part on the cell cycle, we showed that it's this robust bistable switch and this parallel degradation of CDK inhibitor P21 and activation of CDK4 that's important. And the barrier really is this inhibitor P21 that has to be removed in order for this to work. And I should say, you can make a knockout mouse that lacks P21, and there's other barriers being present, and we can also to some degree simulate that in our system. So it's not, this doesn't necessarily mean that this is the only barrier, but in these fibroblasts and many other quiescent cells, we believe this is the primary barrier that is in place to first prevent the cell cycle to start. If you take it out, there might be other barriers that are still there and can help prevent the cell from entering the cell cycle. And in the other part, I showed this modularity that mostly is based on a mechanical motility term that allows cells within a sheet to self-polarize and migrate, and these steering mechanisms that can allow the coordination. And I just want to mention the people who did most of the work. It's Phil Vitorino and Mark Hammer who did the work on the modeling of the migration. Jong-Min Kim has helped them with it. Byung-Wook Park, together with Won-Do Heo, who has now his own laboratory in KAIST in Korea, did most of the work on this cell cycle restriction point regulation. And some of the other people have contributed to this work. Thank you very much.
That was fantastic. I want to go back to your original emphasis of the importance of single cell measurements. Let us set aside some cells which exist as single cells, like hematopoietic cells, or the cells which happen to be in synchrony. In reality, in the real world, there are really no single cells. Cells exist in aggregates. They may be similar or they may be different. So the exercise then becomes that if we have made measurements from single cells, how do we integrate all this modular activation of this or that module to create an aggregate response? Are there tools to do that?
Yeah, I think you're mentioning a very important point. And I should say that in this case of these collective migration responses, there aren't really individual cells. They have mechanical interactions, and they also might have some paracrine interactions. So we think actually the single cell variability, you take something like this cell cycle entry pathway I showed. So the variability, even if you have a primary cell and you look at isolated cell, what the cell variability is huge in terms of protein expression in a cell, in terms of how they respond with a particular output. So we think in a real tissue, the primary synchronization in most cases is coming from secretory or mechanical or electrical synchronization stimuli that help cells that are very close to do something to do it. So I think in many of these cases, the ultimate synchronization, we think, is coming from an external stimulus. So if you see often in these cultures like coordinated calcium activity or so, and this is most likely coming from secreted factors that coordinate that. And there's many in vivo evidences, like in hepatocytes and other, that whole organs are synchronizing a lot of their activity across the organs or in parts of the organ via, in sometimes gap junction connections, but in many other cases maybe by just some secreted paracrine factors.
In your sheath migration movements, the cells in the sheaths are all identical? Because you mentioned that solitary cells do not migrate.
Yeah, they're identical. And but we can make in these cultures heterogeneous culture by mixing two populations. So we can, for example, that's what I mentioned before, we can mix in FGF receptor negative cells with the FGF receptor positive cells and look if they behave different. So we did these kind of experiments in this. But what I showed is they were identical at this time. Yeah.
I have a question related to the collective behavior of those sheets. Have you ever looked at collective behavior in a three-dimensional system? So
we tried some assays, but have so far failed to make them reproducible enough for automated analysis. Yeah, because we would like to do it with some kind of automated imaging to get enough data. We have not been able to do that so far. Yeah.
In the in the collective migration modules, did heterotrimeric G proteins show up anywhere?
So we didn't target them, except of a few specific ones that didn't show up, but we didn't target them systematically. Yeah? So it might many of them might show up, but we didn't see them. Also, in some cases, we find with GPCRs it's difficult to knock them down in 2 days with RNAi. So.